So hello everybody. Nice to speak against a living audience. It's probably not the first to say. Today I will talk about the multi-component acousto-vibratory courtship, which has been described for the first time, to my own fascination as well, in a model cricket species. It's not so much of a data quantification so far. It's more of a story, and I think nice perspectives of it, but I nevertheless hope you will like it. So crickets, we all know, and it's one of the most used invertebrate model to study mating systems and communication at different levels, including behavior from the sensory modalities, acoustics, and its neuronal basis. And for the most in Gryllus, but also Gryllus and Acheta domesticus, the house cricket. So their acoustic signaling has been extensively studied for over 100 years, and I have to mention an important contribution of a Slovenian bioacoustician, which ironically at that time really struggled to prove that crickets communicate via sound and not entirely via the substrate. So there has been some interest, obviously, in their substrate-borne communication some time ago, but especially consider their signals which are emitted as a byproduct of stridulation. But this research ceased almost entirely in the late 80s, and everything was focused to their airborne communication. However, crickets use a large number of other signaling mechanisms that can be related to vibration production, in addition to stridulation, which are very important in their short-range interactions, especially aggression, but also courtship, but have received their very well-known behavior from different families and subfamilies of crickets, but have received surprisingly scarce attention in experimental research. And there was also almost complete lack of signal record, especially within the Grylloidea, the field crickets, and there is the quantitative analysis of this emission. And especially for courtship, which has been extensively investigated now for many years, it became clear that it is clearly a multimodal process, that the female cannot get all information she needs for the choice from the acoustic signals alone. But so far, there has been no real insight into the role of substrate vibration. So about the function of courtship song, it has been shown in various species of field crickets that it has a really low potential for species identification, and it should predominantly serve providing information for the female about the male quality for her choice. But however, this has not been resolved because female often show either no selectivity for individual courtship song parameters, or the females do prefer males that invest more vigor into the courtship singing, like higher rates and duration of song elements, but for the most part, these parameters show no condition dependence. Therefore, one may, of course, presume that female would prefer from analyzing information from other modalities as well. And this is what I didn't mention before. It is generally acknowledged that all field crickets do some kind of tremulation during the short-range courtship of the female, and I said, okay, it may be worth to look at the signals and see whether there is any potential, they have any potential for the female to improve her or to make the courtship signals more reliable. So an example of courtship song here from Acheta domesticus. It is typically composed in field cricket from high and low frequency pulses that occur in repetitive elements called phrases. And in Acheta, there is one high frequency pulse called tick that occurs with a low repetition frequency or with a low rate, and a number of low-frequency pulses, which are also called B pulses, that occur with a higher rate. So here would be how a courtship song and a courtship display would look like from the eyes and ears of a bioacoustician. Sorry. So we can hear the high frequency ticks and between the trill of the low frequency elements, and you can see that the male does some lateral movement that apparently so far nobody found interesting. And I said, okay, I will look for different species of crickets, what this information this may contain. So I have used for this a simple setup. I have put pairs of free crickets into a loudspeaker membrane, recorded their signals with the laser, and soon I realized that vibration does not have much sense without recording sound also, so I had to channel recordings of sound and vibration, recorded the behavior also with the camera, and I conducted everything under the red light because these are nightly active animals, and I also used the same setup in some further experiments. And so, to my surprise, this is what I have found. This is a close-up, and the camera is connected to the vibration input. So this is the cricket that started to court after contacting the female. Opa, sorry. Okay. And you can see it's much more than the courtship song. It's a complex interplay of the song, the lateral body tremulation, and leg drumming. Unfortunately, we don't have the high-speed video camera yet, but it was apparent that the drumming is mostly performed by the foreleg and also a little bit by the mid-leg. But it seems to be a very difficult maneuver to coordinate. So, okay, and then I was stuck with my first experimental species. So this is how the auditory and vibratory recording of the display looks like. So much more complex that may be presumed, and this is how it can be heard when both channels are displayed. So we have the sound and this drum that we can hear, that they come somehow in packages, and the low frequency tremulation, which we actually don't hear at the back. So what is what in this display? These are the high frequency ticks. These are the low frequency stridulation pulses, and then a series of drumming signals that for which we can see that they tend to appear very regularly exchanged or in the pauses between the low frequency stridulation pulses. However, this is not consistent. This is rarely achieved, and most of the, in most cases in this long repetitive display, not all of the low frequency pulses is followed by leg drumming, but some. And then you have this background of almost constant tremulation. And if I filter the tremulation out, this is the drumming signal, which is also, to my extra surprise, very well audible. I could hear it when the crickets are an appropriate substrate, so in this case on a loudspeaker membrane, and their amplitude is at least as high as these low frequency pulses, sometimes even higher than the tick, which is the intenser one. So a few words about the signal analysis. Contrasting the singing, the stridulation, which is immediately in its typical structure. And so you can take from each animal the first, let's say, 15 seconds for the analysis. It is not so with the vibratory components, which are added subsequently into the display. So typically a warm-up of several seconds is needed until every component is displayed and they reach a kind of like steady state. And these segments I have considered for the analysis. However, a constraint in my setup also was that female was freely walking, and often the males, when started to search for her, when she was not in an immediate position behind the male, they turned, and also contrasting the stridulation, which was largely not really disturbed by walking or moving. Every movement of the male, which was not part of the courtship displays, of course, constrained or inhibited its vibratory performance. So despite it had a lot of recordings, surprisingly low amount were actually then useful for analysis, because this drumming and stridulation display was also quite variable. So I took only those examples where I took relatively long sequences of at least 60 to 70 phrases for each individual into account for quantitative analysis. And a few words about the tremulation. As I said, almost continuously a low-frequency signal, with very low variability in the frequency domain. But overall, the structure of the amplitude of modulation was not entirely clear. So what to measure here? But overall, it appears to be, or it was clearly associated with the drumming. So more drumming, more tremulation. However, in some cases, in specific position of the cricket on the membrane, and we have to know that the cricket does the lateral movement while we measure vertical vibration of the substrate. But in specific cases, this tight association between drumming and tremulation was really clear. So every drum was followed by a clear amplitude peak in the tremulation. However, because of this variability, I said I could not evaluate this from the vibration recording. I could generally, for example, evaluate like the effective amplitude for each male from phrase to phrase and compare them, but for this, I would have to have standardized position, both of the signaler and the recording, which was not the case in our setup. So this approach would not work for the analysis. That's why I used the same sections in the video recording and analyzed the movement frequencies from the video. This is what I will correlate with the rate of other signals in the next slides. And just shortly about the drumming, these are on the other hand really short transient signals with modal energy emitted in the first few milliseconds. So the important information of this is in their timing. And they're typically, as usual for any percussion, are very broadband signals with relatively really high peak frequencies, a little bit depending on which part of the membrane the cricket stood, because it was not homogeneous. And maybe for those which consider these frequencies really high for vibrational communication, I could show you some comparison to neurophysiology of cricket vibration receptors. And actually in this range, the cricket detect vibrational signal the best. So now I will show you just first insight into the correlation of the rates and counts of this signal and what can we see from this. And first I have quantified the rates of stridulation in the same sequences as I have analyzed both auditory and vibratory emission. And this is nothing new, but still from these sequences which I considered, I could see that the rate of stridulation, ticks and pauses are correlated. And here what I show are the in-phrase means for the rate B pauses, which are emitted not continuously. So for each of the phrase, I calculated the mean, and these are the mean in-phrase values for 10 males. So the faster the male stridulates, the faster are the ticks, the faster the rates, the B buzzes are produced. And also, if I consider in-phrase drumming rates, I could see that the rate of stridulation and the rate of drumming is also significantly positively correlated. And so the faster the male stridulates, the faster it tries to produce the drumming. And because, as you could see, these somehow tend to be coupled with the pauses between the B pauses. Okay, these are the in-phrase averages. And also the correlation between drumming and tremulation, the latter one from the video records, we could also see that these rates are statistically significantly correlated. And okay, and I said, okay, everything is correlated, so why then the female signal would not choose the males based on the high stridulation rates if these other two components don't bring much new information for her? But of course, if you remember, the rates of drumming follow the stridulation, but they are not persistent throughout the phrase. So this is what I have looked next. How efficient are males in the persistency of the drumming throughout the phrase? So I have expressed this persistency as the ratio between the actual amount of the number of drums versus the number of B low frequency pauses per phrase. And here we could see that this this ratio, this persistency, is negatively correlated to the rate of stridulation. In other words, the higher the higher the rate of stridulation, the less efficiently the male can follow this by the drumming, and this reflects not only the energetic demands, but probably also the difficulty to correlate this at the neuromuscular level, I would say. And these are the individual means which also show this negative correlation. Okay, so this indicates that the combined display could have the potential to honestly reflect the male condition. As we can remember, in Akita domesticus, the female did prefer males in better condition. However, condition was not reflected in the parameter of courtship song. And we can maybe now a little bit more understand why. And here's another example from Grylloidea persynvalicus, when they show that the females do select for the higher stridulation rate, but these rates can be produced both by the low and high quality males, which were on the low or high diet. But if case this is the complex display, as I have found now in Akita, the females would have much more information to distinguish between the high and low quality males. So this is what I would have to look, not about the condition dependence, not only of this signaling persistence of all the components through the phrases, but also of the degree of signal synchronization. And this is what I would still need to analyze from my current data set, because I have noted that some males, even though that they're drumming a lot, they are really bad in timing of their drums in the poses of stridulation, of short stridulation pauses. But some are better. So this would be another view that the females may assess, which would not reflect the male condition, but its genetic quality reflected in the ability of the male to coordinate these movements in a highly synchronized manner. So the conclusions be that this is the first significant insight with the quantitative analysis of the vibrational signals in the courtship of field crickets, where I have found that not only a courtship song with some tremulation, but the complex multi-component acousto-vibrational display with stridulation, drumming, and tremulation is present. And in the future, the condition dependence and putative female preference would have to be tested for the specific elements and the combined display. And I actually have already done this, but I figured out there will be no time to show this on today's talk. And in the light of this data, the courtship behavior of other field crickets need to be reinvestigated with a focus on the vibrational channel, which may increase a much more understanding of their courtship process, and also make the crickets even more relevant as the study system of multimodal communication, not only combining acoustic and chemical signals, which is currently intensively considered, but also, and maybe even predominantly, the complementary law and some vibration, which are related not only functionally, but also evolutionary much more closely, that are chemical and auditory signals. So this is all.